When you perform a DNA sequencing, the obtained result is correct. How do we decide which gene or genes to analyze in a patient? Exactly. We need both clinical information and the mode of inheritance in the family in order to be able to determine which gene or genes that are the most likely candidates for the disease-causing gene in this specific family. Each inherited characteristic or disease has a mode of inheritance, depending on where the gene is located, and whether the mutation causes disease if you have it on just one allele, or if you only see the phenotype if all alleles are mutated. We use the term genotype to describe the DNA sequence of an individual. What is then the phenotype? Correct. The phenotype is defined by the observable characteristics in the organism. For instance, your phenotype includes the color of your eyes or your hair, but it's also whether you have any diseases, how well you respond to medication, etc. In the case of genetic disorders, the phenotype is the expression of the disease. What is the relationship between genotype and phenotype? You are absolutely right. The genotype, the unique combination of alleles, is a major influencing factor of the phenotype, however, is not the only one. Phenotypes are influenced by environmental factors, thus although identical twins have the same genotype, they can have different phenotypes. Also, many different genotypes can produce the same phenotype. Phenotypes are not directly inherited. Alright, time flies when you are having fun. It is already time for the morning conference. Listen carefully to your colleagues, there is always something new to learn from other people. Welcome to the morning conference. This is a meeting where all the doctors in the medical genetics department discuss their most recent cases. Our aim is to help each other understand our cases and to give the best advice to our patients. Pedigrees are a very useful tool for genetic studies. Are you familiar with the symbols used in a pedigree? What does it mean if a person is depicted as an open circle with a line across it? Check out the theory if you want to refresh your knowledge first. Correct. In pedigrees, women are represented as circles, and men are represented as squares. A line crossing these symbols means that the person died. A filled symbol means that the person is or was affected by the disease under study. When counseling families with inherited disorders, we are often asked as to what is the risk that a specific individual, e.g., an unborn child, will develop the disease. In order to do a risk calculation, you need to know the mode of inheritance of the disease and the pedigree of the family, and you need to know a bit about calculating probability. You can access a brief risk calculation crash course by clicking the View Theory button. As discussions of disease risk is an important part of this morning conference, let's make sure that we all know the basics. Let's go through some examples of risk assessment in the most frequent modes of inheritance. In this family, the mother is affected with an autosomal dominant inherited disorder, and we therefore expect her to be heterozygous for the disease-causing mutation. What is the risk that the unborn child has inherited the disease? Correct. We inherit one chromosome from each homologous chromosome pair from each of our parents. As patients affected with an autosomal dominant disorder most often are heterozygous for the disease-causing mutation, we expect that the affected mother has a 50% risk of passing on her mutated gene allele to her child. If the child receives the mutation, they will in most cases develop the disease. In disorders with autosomal recessive inheritance, the affected children have received a disease-causing mutation from both parents. In the family shown in the pedigree, we can conclude that the parents are both healthy, heterozygous carriers of the mutation. What is the risk that their second child will develop the disease just as their first daughter? Exactly. Individuals affected with an autosomal recessive disorder are homozygous. They carry a disease-causing allele on both chromosomes. If a person is heterozygous, we expect that they are a healthy carrier and not affected. When you calculate the risk that a child has inherited the disorder, you assess the risk that the mother passes on a mutation, and multiply this with the risk that the father passes on a mutation. In the case where both parents are healthy carriers, each have a 50% risk of passing on the mutation, and the disease risk is 50 times 50 equals 25%. If a family member suffers from an autosomal recessive inherited disorder, the carrier risk of the following generations is much higher than what is found in the general population. In the pedigree, you see an example of a family with an autosomal recessive disorder. A young, healthy couple are expecting a child. The carrier risk of the father depends on how frequent this disease is in the population from which he originates, but the risk that the mother is a healthy carrier can be calculated from the pedigree. What is the mother's risk of being a carrier? Very good. When assessing the risk that a sibling of an individual affected with an autosomal recessive disorder, we use our knowledge of inheritance to conclude that both parents must be heterozygous carriers. The person whose risk we are assessing cannot be homozygous for the mutation, as she is unaffected. When calculating her carrier risk, we can therefore exclude the genotype where both parents passed on the mutated allele, leaving these three possible genotypes: a mutated allele from mother plus an unaffected allele from father, an unaffected allele from mother plus a mutated allele from father, two unaffected alleles. This means that in 2 out of the 3 possible genotypes, the unaffected sibling is a heterozygous carrier, so the risk is 2/3. In this family, a young man is affected with an X-linked recessive disorder. In these disorders, we often see more affected men than women, as a man with a mutation on his X chromosome will be affected, whereas a woman heterozygous for the mutation will be an unaffected carrier. In this case, the mother of the young man must be a heterozygous carrier of the disease, as he has inherited his X chromosome with the mutation from her. What is the risk that the man's sibling and child will develop the disease, provided that the man's wife is not a carrier? Correct. When assessing the risk of disease in families with X-linked recessive disorders, it is important to follow the X chromosomes. Remember that a father passes on his X chromosome to all of his daughters and to none of his sons, as they get his Y chromosome. The child will not develop the disease, as girls will receive the mutated allele on the X chromosome from their father, but will also inherit an unaffected X chromosome from the mother, and boys will not get his X chromosome at all. In the case of the sibling, indicated by number 2 in the pedigree, we know that the mother of the young man must be a carrier, and the man's siblings have a 50% chance of inheriting the mutation. If the man has a sister, she has a 50% risk of being a carrier, but a 0% risk of the disease. If he has a brother, has a 50% risk of disease. This is the pedigree of a family affected by a connective tissue disorder. What is the expected mode of inheritance of the disease? Remember that affected family members are represented as filled-in shapes and unaffected family members by open shapes. Superb. The expected mode of inheritance is autosomal because both men and women are affected. It is dominant because it appears in every generation. The autosomal dominant mode of inheritance and symptoms found in the patients from this family indicate that they suffer from Marfan syndrome. It is an inherited disorder caused by mutations in the FBN1 gene. What is the most feasible way to investigate if this is the correct diagnosis? Since alterations in the FBN1 gene are the cause of Marfan syndrome, sequencing of that gene in the patients is the most efficient way of revealing if they suffer from this condition. We expect the affected family members to be heterozygous for a mutated allele of the FBN1 gene. According to Annabelle, there is a wide range in the severity of the symptoms between the family members. Can such a variation occur in an inherited disorder? Impressive. You are right, even among patients carrying the same mutation, different phenotypes can be observed. The reason for this can be that other genes, or the environment, have influence on the phenotype. That's it from my side for now. Go ahead and have a chat with the doctors in the morning conference and learn more about their most recent cases. Pay attention because you never know which of the reported cases would be your next patient.
Click on any of the highlighted doctors to learn about a new case. A young couple, Sarah and David, have just had their first child. Sarah and David are cousins, and their grandmother had a sister who died of Canavan disease. Canavan disease is a serious metabolic disorder with an expected lifespan of 10 to 15 years. The disease is particularly frequent in some populations, e.g. Ashkenazi Jews, where 80% of the affected are homozygous for the mutation E285A. Both boys and girls are affected with equal frequency. Based on these informations, what is the mode of inheritance of Canavan disease? No, this is incorrect. Please try again. You are right. The fact that boys and girls are equally affected indicates an autosomal mode of inheritance. The fact that patients have been shown to be homozygous for a certain mutation suggests a recessive mode of inheritance. Based on the pedigree, what is the risk that the baby, indicated by V.1 in the pedigree, suffers from Canavan disease? How is it calculated?
Ayan. Sandali lang ha.
Kunin mo nga.
Ano yung katawan paluan?
Exactly. Since the grandmother had a sister with the disease, her parents must both have been heterozygous carriers. As the grandmother was not affected, she can have 3 possible genotypes: carrier of the mutation from her mother, carrier of the mutation from her father, or homozygous for the unaffected allele, resulting in a 2 in 3 carrier risk. With each generation, there is a 1 in 2 chance the mutation is passed on, resulting in a 1 in 6 chance for both David and Sarah to be carriers. To calculate the risk that the daughter of David and Sarah have 2 disease alleles, you need to multiply the risk that David passes on the mutation with the risk that Sarah does. 1 in 6 by 1 in 2 by 1 in 6 by 1 in 2 equals 1 in 144. We have sequenced the baby's ASPA gene and tested for the frequent mutation E285A. The girl does not carry this mutation. What can we conclude from this? You are right. The fact that she does not carry the most frequent mutation doesn't allow us to discard the hypothesis of a less common mutation leading to Canavan disease. Thanks a lot for your comments. Click on any of the highlighted doctors to learn about a new case. Mary is pregnant, and she and her husband are seeking counseling as there is at least 1 case of suspected Fabry disease in Mary's family. Her mother's brother suffered from severe pains in the limbs, was unable to produce sweat, and died due to kidney failure at the age of 47. Mary has a nephew, age 7, who presents with numerous vascular cutaneous lesions, and the boy's mother has a few of these lesions too. These lesions may be a sign of Fabry disease, but they appear as part of other disorders too, or in healthy individuals. If we consider the worst-case scenario, where Mary's nephew has Fabry disease, then what is the most likely mode of inheritance in the family? You are right. The mode of inheritance is recessive, either X-linked or autosomal. However, it is the most likely X-linked recessive. What is the risk of Fabry disease in Mary's unborn child, provided that her nephew does have Fabry disease, and how is it calculated? If Mary's nephew suffers from Fabry disease, we expect his mother, Mary's sister, to be a heterozygous carrier of the mutation. Mary's risk of also being a heterozygous carrier of Fabry is 1 out of 2. To her child, Mary passes on one of her X chromosomes, either the healthy one or the one with a 1 out of 2 risk of mutation. The risk that the child's X chromosome from Mary has the mutation is 1 out of 2 times 1 out of 2 equals 1 out of 4. Only a boy will have Fabry disease, so we need to multiply by 1 out of 2 again to account for a 50% change of the child being a boy. Male patients suffering from Fabry disease have mutations in the alpha-galactosidase gene, an enzyme activity of alpha-galactosidase between 0 and 12 units. Healthy individuals have enzyme activities of more than 75 units. What type of mutation do we expect to find in the alpha-galactosidase gene? The reduced enzyme activity of alpha-galactosidase in Fabry patients indicate that they carry a mutation in the gene encoding alpha-galactosidase, and that this mutation renders the enzyme less active, or inhibit its production. These mutations are collectively referred to as loss-of-function mutations.
Well, thanks everybody for coming to the morning conference. It has been as interesting as usual. Now back to work. Good luck with your consultations today.
Meet SIF and Anton.
Hello, doctor. I'm here with my wife SIF and need some advice from you.
Good morning. My name is Frank and I am a medical genetics doctor working here at the medical genetics department. How can I help you?
Hi, Dr. Frank. I am SIF, and this is my husband Anton. I am pregnant with our first child, but we've started to worry. My mother just told me that my father, who died last summer, had an aunt who died as a child from cystic fibrosis, and that really scared me. She was the younger sister of my father's father, but they are all dead, so I can't ask for more information.
We saw a television program about this family with twins suffering from cystic fibrosis, and these kids had serious lung infections and lots of other bad symptoms. The parents in the program didn't know they were at risk before the twins were born. And a doctor was interviewed, and he said that 1 in 30 Danes carry a mutation that can cause cystic fibrosis, and we are both from Denmark. I don't know much about my family, except that my father was adopted. My mother has no contact with her relatives, so I just don't know if there are any cases of cystic fibrosis in my family. What can you tell us about the risk that our baby has cystic fibrosis?
What is the first thing to do when assessing the risk of a fetus suffering from an inherited disease? Indeed, in order to judge the risk of a certain inherited disorder in a fetus, we need to look at how the disease is occurring in the future parents' families. To do so, we need to draw the family pedigree. What we are going to do is to draw your pedigree, then I will be able to assess the risk. Do you have any siblings, Anton?
Not me, but SIF has an older brother.
Click on the PC screen and draw the pedigree of SIF and Anton. Look at the screen for instructions or raise the lab pad for more information.
Well done. You successfully created the pedigree for Anton and SIF. When studying a disease with an autosomal recessive mode of inheritance, how do you know if a person will develop the disease based on his or her genes? We assess the genotype of the relevant individuals to see if they are homozygous for disease-causing mutations in the gene related to the inherited disease. We expect these to develop the disease. We also look for healthy carriers that are heterozygous for the mutation, as these may pass on the mutation to their offspring. Can you calculate the risk that the fetus will develop cystic fibrosis? What is the risk that SIF is carrying the mutation? Exactly. We start with Sif's father's aunt, who had cystic fibrosis. As the mode of inheritance is autosomal recessive, she must have been homozygous for cystic fibrosis mutation, and her parents therefore must have both carried a mutation. Since Sif's grandfather was healthy, he can't have been homozygous too, leaving three options for his genotype: heterozygous with a mutation from his mother, heterozygous with a mutation from his father, or homozygous with two unaffected alleles. Hence, in 2 out of 3 options, he's a heterozygous carrier. Every time this risk is passed on to offspring, it is reduced to half the risk. If Anton's carrier risk is 1 in 30, and that of his wife is 1 in 6, then what is the risk that the unborn child will develop cystic fibrosis? Correct. You calculate the risk that the child is homozygous by multiplying the risk that the mother passes on a mutation with the risk that the father does so. Analyses are needed to tell whether the unborn child will develop cystic fibrosis. Exactly. As research has shown that cystic fibrosis is caused by mutations in the CFTR gene, we only need to assess the sequence of this gene in the fetus. If you analyze the genotype of an autosomal recessive gene in the fetus, will that give you information about the genotypes of the parents? Good point, as we cannot always be sure about the genotypes of the parents based on the results from analysis of the fetus. It will be a good idea to analyze the parents too. Then we can assess the risk for future children of the couple.
Thank you very much. This is a difficult situation for us, and having a doctor who patiently answers all our questions is very helpful.
Hi, glad that you are back to the lab again. We have received the blood samples from SIF and Anton and the Corian villus sample from the fetus. I have extracted and purified the DNA from them. I have also provided you with a DNA sample from a patient carrying the F508del mutation. This is the first mutation in the CFTR gene that we investigate when there are suspicions of cystic fibrosis. If you live in Denmark but you are not Danish, do you have the same probability of carrying the F508del mutation in the CFTR gene? Brilliant. It is your genetic background that determines the probability of carrying certain mutations. You will have to read the literature to find what are the predominant mutations in your genetic background. For example, in the US only 60% of the disease-causing alleles are the F508del, and almost 10% have the R117H mutation, which is very rare in Denmark. What would you do first if your goal was to investigate the presence of a mutation in a DNA sample? Exactly. To investigate if a mutation is present in a certain DNA sample, you need to amplify the region of interest using PCR. Go to the PCR workbench. Click on the glove pack to put them on. Pick a fresh PCR tube from the pink bag and put it in the rack. Add the DNA from the fetus, the PCR mix and the primers for the F508del mutation using the pipette. You will find all of these on the workbench. Hover over equipment and reagents to find the correct ones. Why are you selecting these primers? Correct. Since both parents are Danish, we will start by looking for the mutation with a higher probability in the Danish population. You should do the same now with the rest of the samples, but you are in a virtual lab so we can do that for you. You will find the samples in the PCR machine, ready for you. Now place the tube in the PCR machine and start the PCR. Here's the sequence of an allele with the F508del mutation, compared to a healthy control. The reading frame of the gene is ATC followed by TTT, which is the codon for phenylalanine, also designated with F. How does the F508del mutation change the CFTR protein? Correct. The mutation is a deletion of 3 nucleotides, which only changes 1 amino acid in the protein, while the rest of the gene is translated correctly. The severe phenotype of the mutation tells us that this phenylalanine is very important for the protein. Research has shown that deletion of it leads to misfolding and degradation of the CFTR protein. Do disease-causing mutations in proteins always lead to loss of function phenotypes? Indeed, sometimes mutations lead to the production of non-functional protein, or to no production of the protein at all, which in the end will lead to loss of the function of that specific protein. However, in some cases the mutation causes a change of the structure, which may lead to both enhanced function of the protein, or that the mutated protein gains new and potentially toxic functions. Which reagent is missing in this list for a PCR, DNA template, buffer, nucleotides and primers? Exactly. Without the DNA polymerase, the PCR can occur because this enzyme is responsible for the synthesis of the new DNA strand. Excellent. With all you know already, make a hypothesis. Will the PCR product of a sample with the mutation be longer, shorter, or the same size as the PCR product of a healthy sample? Good. All hypotheses are valid and need to be proven right or wrong. However, in order to make a hypothesis, it is a good idea to review at all the information we have available. This can be reading the bibliography or even asking your colleagues whether they have had the same question before. In this case, we can have a look at the PCR primers and the part of the CFTR gene to which the primers bind to and calculate the theoretical size of the PCR product. Go to Workbench 3 and click on the PC screen. Wait for the PC to start up. Take a look at the following DNA sequence. Please find the right position to place forward and reverse primer and press submit. Submit the correct forward and reverse primer position. Click buttons 1, 2 or 3 to see the options highlighted on the DNA sequence. Click submit when you are done. Unfortunately, that was not correct. Please try again. Awesome. We now know that the PCR product from the unaffected allele is 84 base pairs. How long do you expect the PCR product from patients with the F508del to be? Excellent. This means that your hypothesis was right. The PCR product of a sample with the F508del mutation will be shorter than the PCR product of a healthy sample. This is good news. Now we know what to expect from our results. Super. We know now that the F508del mutation will lead to a shorter PCR product than the controls carrying the wild-type sequence. Let's analyze the length of our samples performing a so-called fragment analysis. Nice. Put the tube back into the PCR rack. Marie will help you with the other tubes. For the fragment analysis, we will use capillary electrophoresis, a more sophisticated electrophoresis machine than the one you saw during the tour in the lab. Bring the PCR tube rack to the electrophoresis workbench. Go to the electrophoresis workbench. Place the PCR tube rack on the workbench. Pick a fresh 1.5 milliliter tube and put it in the rack. Use the pipette to add the size standard. Add the size standard to the empty 1.5 milliliter tube. Use the pipette to add some formamide. Add formamide to the tube containing the size standard. Add PCR product from the fetus. Denature the PCR product mix at 95 degrees Celsius in the thermoshaker. Well done. In a real lab, you will have to do the same with all your samples. But you are lucky to be in a virtual lab and that it has been done for you already. You can find the tubes ready for the electrophoresis in the capillary electrophoresis machine. Add your tube there. Take the tube out of the thermoshaker and put it in the capillary electrophoresis machine. Close the capillary electrophoresis machine and press start. Now the capillary electrophoresis machine will take the sample from the tube and inject it into a capillary, a very thin tube filled with polydimethylacrylamide, the polymer where the separation will take place. You can learn about the principles of electrophoresis by clicking in the View Theory button. In an electrophoresis experiment, molecules are separated according to which characteristic? Exactly. Larger molecules are retained for a longer time in the matrix while smaller molecules move much faster. This difference is the base for the separation. How can we estimate the size of DNA fragments? You're right. By adding fragments of a known size in the DNA ladder or size standard, we can estimate the size of our product by comparing the bands to the bands of the known fragments. Great job! Now the electrophoresis has finished. You can analyze the results on the computer. It has been a pleasure working with you. Go to the computer on the workbench 3 and click on screen to analyze your results. Now you will be asked several questions. Click Explore to go back to the computer screen if you need to have a look at your results to answer the questions. Click on the sample and PCR name to view a graph showing the fragments detected. What is the genotype of the fetus? Yes, the analysis of the fetus DNA displays two PCR fragments of different size, showing that the fetus carries one allele with the F508del mutation, and one unaffected allele. What is the genotype of SIF? Yes, the analysis of DNA from SIF reveals that she, like the fetus, has one unaffected allele and one allele carrying the F508del mutation. What is the genotype of Anton? Indeed, the analysis of DNA from Anton reveals that he, like both SIF and the fetus, carries one unaffected allele and one allele with the F508del mutation. The senior doc just went online. Click on the middle icon on the PC screen to contact him.
If we have been closer in age, it would have been fine.
Have you reached a conclusion? Based on the results, will the fetus develop cystic fibrosis? No, you are not correct. This is very important for the future of the fetus and the parents, so think it over once again. Excellent. The fetus is heterozygous for the disease causing F508del mutation, but it also carries an unaffected allele of the CFTR gene, meaning that it will be able to produce normal CFTR protein. The child will therefore not develop cystic fibrosis. What would have been the fate of the fetus if both parents had passed on the F508del mutation? Excellent. You are ready to give genetic counseling to SIF and Anton. Go to the consultation room. They are waiting for you.
Welcome back. I have good news for you. Your child will not develop cystic fibrosis. We found that the fetus is expected to be a healthy carrier, as it has one copy of a mutation that may cause cystic fibrosis, but also one unaffected allele of the CFTR gene. However, we need to talk about the future, as our analyses revealed that both of you carry this mutation called F508del. It is the most common mutation in the CFTR gene, but unfortunately it is also a mutation leading to a very severe form of the disease if you have two copies of it. Let's talk about this some more. You must have a lot of questions.
Oh, that is great news. I am so happy that our future baby will be healthy.
What factors might influence the severity of cystic fibrosis caused by the F508del mutation? In some inherited diseases, including cystic fibrosis, there is a genotype-phenotype correlation. This means that different mutations in the gene lead to varying severity of the disease. Other genes and the environment can also have an impact. Both Anton and SIF carry the F508del mutation, which causes severe cystic fibrosis. Why do they not suffer from cystic fibrosis? Cystic fibrosis is a recessive disorder. You need to be homozygous to actually suffer from the disease. Most recessive disorders, as cystic fibrosis, are caused by loss of function of the mutated genes, meaning that the patient is not able to produce functional protein, or produces way too little. In your case, the unaffected allele you carry is able to produce enough CFTR so you don't develop the disease. What is the risk for any future children of SIF and Anton to inherit two disease-causing alleles from them? Exactly. We now know from the laboratory analyses that both of you are heterozygous for the F508del mutation. Each of you has a risk of 1 in 2 of passing on this mutation to your offspring. Fortunately, it takes 2 mutated alleles of the gene to cause a child to become ill, so we calculate the risk that both of you pass on the mutation to the same child, 1 in 2 by 1 in 2 equals 1 in 4. Given that 88% of the cystic fibrosis mutations in Denmark is the F508del, what is the most likely explanation why one mutation is so frequent in the Danish population, when it is not as frequently found in other populations? That's it. It is not likely that the F508del has skipped negative selection, as this is one of the most severe mutations. And all families suspected of having cystic fibrosis will be examined until a mutation is found. I hope this answered all of your questions for now. In case you plan on having more children, and want prenatal analysis of the fetus, we could offer you pre-implantation genetic diagnosis where we perform in vitro fertilization and test the genotype of the embryos before implanting them. Please don't hesitate to approach me with any questions you may have in the future. Good luck.
SIF and Anton had a healthy daughter. SIF is now pregnant with a healthy boy, genotyped using pre-implantation genetic diagnosis. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress.
Okay, so that's for the last simulation for the part 2. Okay. Now we will be having our post lab discussion on this part 2, 3 labster simulations.